In this particular video, we are going to discuss about the membrane transport, how polar and non-polar molecules are transported across the membrane. So, let's start with what is membrane transport. As we all know about the biomembrane, which is a bilayer composed of phospholipids and proteins. These phospholipids and proteins form a selectively permeable barrier which separate the cell from outside. It means this bilayer of phospholipids and various type of proteins which are present in the biomembrane act as a barrier between the cytoplasm and its surroundings. This selective permeability allows the membrane to control the entry and exit of various substances which can be polar, non-polar, also this selective permeability maintain the internal conditions which are necessary for the cellular processes. Okay, then the membrane transport is essential to transport essential molecules such as nutrient, gases, water inside the cell to facilitate various cellular processes. By regulating ionic and molecular concentrations, membrane transport preserves the cellular volume, osmotic balance, and biochemical stability, which is critical for the cell survival. Okay, then membrane transport also ensure that there is a timely supply of various important substances which are required for various reactions, or the removal of harmful substances, or we can say the byproducts which are not useful in that process, to be successfully removed out of the cell. Okay, also the selective transport function maintain the cellular identity, facilitate immune recognition, and allows the cell to adapt and respond to the environmental changes. Okay, now the membrane transport is of two types. The membrane transport is of two types. First is passive transport and second is active transport. So the main difference between the two is passive transport requires no energy expense. Okay, and active transport require energy in the form of ATP. Molecules are passively transported without the expense of energy, but in case of active transport, ATP is used to transport various molecules. Okay, another difference is that in passive transport, molecules are transported toward the concentration gradient. Toward the concentration gradient, but in active transport, the movement of these molecule is against the concentration gradient. Concentration gradient, and that's why energy is used, because we cannot transport any molecule against the concentration gradient without using energy. Okay, passive transport includes osmosis, simple diffusion, and facilitated diffusion. In active transport, we have primary active transport, secondary active transport, and vesicular transport. Let's discuss one by one. First, the passive transport. As we have already discussed, the passive transport moves the molecule across the cell membrane without the use of cellular energy in the form of ATP. We already know that passive transport doesn't require any energy, and it is totally dependent on the concentration gradient or the electrochemical gradient to drive the movement of those molecules. Second, these substances are moved from the region of higher concentration to the lower concentration. That's why no energy is required, because we are flowing with the flow, okay, from the higher concentration to the lower concentration, and the movement of molecules will continue until a balanced condition is attained at the both sides. Okay, there is an equilibrium condition in both the sides. Passive transport includes the mechanisms such as simple diffusion, where small non-polar molecules, please remember, in simple diffusion only small non-polar molecules like oxygen, carbon dioxide pass directly through the lipid bilayer. Okay, polar molecules cannot pass through the membrane, okay, through simple diffusion or osmosis. More selective transport occur facilitated diffusion. Facilitated means we require a carrier for the transfer of molecules from extracellular matrix to the cytoplasm, or from the cytoplasm to the extracellular matrix, where specific transport proteins are required as carrier molecule. This can be channel proteins or these can be carrier proteins, which transfer or which moves hydrophilic or larger polar molecules. Yes, in simple diffusion we can only transport non-polar molecules, but in facilitated diffusion these channel proteins or carrier proteins can carry large molecules, large polar molecules as well. Okay, the first type of passive transport is osmosis. In osmosis, please remember, there is a transport of there is a transport of solvent molecules, solvent molecules from higher concentration to lower, but through a semi permeable membrane, sorry, through what's going on here, just wait a second. Something went wrong here, so we can write it here, through a semi permeable membrane. Okay, so I will show a video, then we will start the lecture. See, in this particular region the amount of solute is less, and in this region the solute concentration is high. Okay, but on the other hand, in this side solvent concentration is high, solvent concentration is high, and on this side solvent concentration is low. So in osmosis, the water molecules will move through a semi-permeable membrane. So we will put a semi-permeable membrane here. Okay, suppose it is a semi-permeable membrane, and the molecules of solute will pass through this semi-permeable membrane towards the region of uh, toward the region where the solute concentration is high but the water concentration or the solvent concentration is low. Okay, so like this, the water molecules are going through the semi-permeable membrane toward the side where the solute concentration is high but the solvent concentration is very low. So we can say the water molecules are transported through a semi-permeable membrane from a higher water potential or the region where the solute concentration is very low to a region of higher solute concentration or the region where there is lower water potential until the equilibrium is achieved. Once equilibrium is achieved both the side, no transport of molecules will carry out. This is an image of reverse osmosis where the movement of solvent is taking place through the semi-permeable membrane from a region of higher solute concentration toward the low solute concentration. Okay, so let us discuss biological membrane regulate osmosis via selective permeability. Okay, mainly through the phospholipid bilayer and aquaporine channels. Aquaporins are those membrane channels. Aquaporine are those membrane channels which help in transportation of water molecules only. Okay, water molecules. Okay, there is another important thing that the driving force of osmosis can be quantified as the osmotic pressure. Some pressure is required to carry out the process of osmosis, and this pressure is known as osmotic pressure, and this pressure is given by pi is equals to ICRT, where pi is the osmotic pressure and I is ionizing constant, C is the molar concentration, R is the gas constant, and T is the absolute temperature. Okay, then on the basis of the external solution, for example, this is a cell. Okay, there is the nucleus in the middle and this cell is placed inside a solution. Okay, there are two types of environment, one which is present inside the cell and other which is present outside the cell, or we can say the external solution. Okay, so on the basis of this, on the basis of external solution, on the basis of external solution, cell experience distinct osmotic conditions like isotonic, hypotonic and hypertonic. Okay, and each of these condition has specific effects on the animal cells as well as the plant cell. Okay, let us discuss these effects one by one. First, isotonic condition. Please remember, iso means same. Okay, and from tonic you can remember solute. Okay, and we are talking about the external solution, right? On the basis of external solution. What's going on here? We are talking about the external solution. Okay, so in isotonic condition, the solute concentration will be equal to that of the cell cytoplasm. It means the external solution will have the same amount of solute present as present inside the cell, or we can say which is present inside the cytoplasm. Okay, so the net movement of water will be zero, because here the solute concentration and the solute concentration of outside is equal, so no net flow of water, so the animal cell will be same, no effect on the animal cell. Then if a plant cell is placed inside the isotonic solution, same, no net flow of water will be there, but the cell becomes flaccid. Okay, so an isotonic solution has a solute concentration equal to that of the cell cytoplasm. Okay, external solution solute concentration will be equal to that of cell cytoplasm solute concentration. Okay, so there will be no net flow of water. Then the effect of animal cell was, uh, for example, we place a erythrocyte or RBC inside an isotonic solution, it will remain normal in size and shape. Okay, there will be no swelling or shrinking. ठीक है, उसके बाद what will be the effect on plant cell? Plant cell also will maintain the equilibrium in isotonic solution, but it will become flaccid, which is a condition intermediate between turgid and plasmolized state. Okay, then hypotonic condition. From the word hypo, you can remember less, and tonic you can remember solute. Okay, so there will be less solute outside than the interior of the cell. Okay, hypotonic solution has a lower solute concentration than the cell interior. As a result, water enters the cell by osmosis. Okay, so we can write here less solute, and inside the cell more solute is there. Okay, if there is less solute outside, then more water concentration will be there, and inside the cell there will be less water concentration. So we have already discussed that uh, transport will occur from the higher concentration toward the lower concentration. So the water molecules will enter the cell because there is less water concentration inside the cell, and when water enter inside the cell, it will when water molecules will enter the cells, either the plant cell or the animal cell, it will make it swell, and sometimes if the osmotic pressure is very high, it will burst the cell. Okay, but in case of plant cell, it only becomes turgid because there is a strong or rigid cell wall around the cell which will prevent it from bursting. The same content is written here. Lacking cell walls, animal cells swell as water enters. If the osmotic imbalance is extreme, the cell may burst, a process known as hemolysis in red blood cell. For example, when RBCs are placed in distilled water, they swell rapidly and eventually rupture, releasing hemoglobin into the medium. And I have already told you that the plant cell become turgid in hypotonic solution. As water enters, the vacuole expand and presses the protoplast tightly against the rigid cell wall. Then hypertonic conditions. Hyper means more, tonic solute. So the concentration of solute will be high outside the cell than the cytoplasm. Okay, so we can write this is external environment. Solute concentration is high, then water concentration will be low here, and water concentration is high inside the cell and solute concentration is low. Water will flow from the higher water concentration toward the low concentration which is outside the cell. Then this cell, animal cell, will shrivels, as well as the plant cell will become plasmolized. Okay, it means the water loss causes the protoplast to shrink away from the cell, and this process is known as plasmolysis. Okay, similarly, if a plasmolized cell is again placed in the hypotonic condition, it will regain its structure. Okay, then it will known as the deplasmolized cell. Here is a diagram. Animal cell in isotonic solution will be normal because there is no net flow of water. In hypotonic solution, the cell become lysed, and in hypertonic it will become shriveled. The plant cell in isotonic solution will become flaccid. In hypotonic solution it will become turgid, and in hypertonic solution it will become plasmolized. The same is written over here. Then next is simple diffusion. Osmosis. In osmosis, there is movement of solvent from higher concentration to lower concentration, but in simple diffusion there is transport of gaseous molecules. Okay, or we can say that small non-polar molecules. Small non-polar molecules. In simple diffusion, the molecules are directly passes through the phospholipid bilayer of the cell membrane without using any type of energy or the transport proteins. These molecules are moved from an area of higher concentration to the low concentration toward the concentration gradient until the equilibrium is attained. Okay, this is a concept. Okay, then only small and non-polar molecules, or we can say lipid soluble molecules such as oxygen, carbon dioxide and nitrogen, can easily diffuse through the membrane by this mechanism. and these are the factors which affect the diffusion rates, which is molecular size, temperature, concentration gradient, and membrane permeability. We will discuss in the next slides. So this is diffusion. There is a high concentration of molecules outside the cell as compared to the inside. Okay, so these molecules will cross the phospholipid bilayer and will move until equilibrium is attained. Okay, here you can see the molecules are moving. Here there is high concentration of molecule, and we can say here is low concentration. So diffusing molecules are diffusing in all the direction toward the low concentration gradient. Okay, आपने ये भी देखा होगा कि sometimes we spray the perfume inside a room in one corner. Those molecules will transfer very fast to the other end. Okay, we can uh, smell that fragrance in the other corner also. So this is the diffusion process. This is due to diffusion of molecules. Then these are the factors which affect the diffusion. Molecular size. So smaller the molecular size, high diffusion will be there. Okay, then non-polar molecules cross easily. Ions cannot penetrate the membrane. Lipid solubility, steroid hormones being hydrophobic, because these hormones are hydrophobic, these can rapidly diffuse through the plasma membrane composed of phospholipid bilayer. Okay, hydrophilic substance cannot cross easily through this membrane. Then membrane fluidity, unsaturated fatty acid and less cholesterol increase fluidity, because the saturated fatty acid decrease fluidity, because saturated fatty acid composed of all the single bonds, so they make the membrane very stable and decrease the fluidity. On the other hand, unsaturated fatty acid have double bonds between them, and due to these double bond, kinks are produced. Okay, we know the structure of phospholipid. There is a glycerol head and fatty acyl chain, and if this fatty acyl chain is made up of uh, unsaturated fatty acid, it will not make the membrane as stable as formed by the saturated fatty acid. Okay, due to kinks produced. So what are kinks? So kinks are basically the bends in the fatty acyl chain produced due to the presence of double bonds. Okay, so we can say unsaturated fatty acid make the membrane or fluidity increase the fluidity of membrane. Okay, then concentration gradient, steeper gradient yield faster diffusion. Okay, and the surface area, more the surface area, more will be the diffusion. Then there is facilitated diffusion. It means it require something to transport molecules from extracellular matrix to the cytoplasm. Okay, most probably in facilitated diffusion, large polar molecules are transported. Okay, so facilitated diffusion is a type of passive transport where molecules move across the cell membrane with the help of specific carrier or the channel proteins without using the cellular ATP. Okay, so there will be either the channel proteins will be there, or specific carrier protein will be there. In this particular image, you can see that this is a channel protein through which the molecules are transported uh, from extracellular matrix to the cytoplasm. In extracellular matrix, here you can see there is high concentration of solute, or we can say whatever these molecules are, they have higher concentration outside the cell as compared to the inside. Okay, so these molecules may be polar or large molecules which require the presence of proteins in the phospholipid bilayer, which will help to cross this membrane. It enables the transport of large polar molecules or charged molecule such as glucose, amino acids and ions, most probably polar molecules that cannot easily diffuse directly through the lipid bilayer. Then channel proteins form hydrophilic pores, and these hydrophilic pores allow these ions or polar molecules to pass rapidly. Okay, while carrier protein undergo conformational changes to pass these substrate across the membrane. Okay, there will be a conformational change in the carrier protein. Okay, this process is also driven by concentration gradient. Okay, movement from the higher concentration toward the low concentration. Then what is the general mechanism of facilitated diffusion? The first process is the binding of solute molecules with these channel, channel proteins or carrier proteins. Then there will be a conformational change in the carriers and gating mechanism in the channels which permit the translocation. In this image you can see this is a channel protein. This is a channel protein. The solute concentration is high outside the cell and here is low. So these molecules are transferred or transported from the higher concentration to the low concentration through this particular channel. Okay, this is based on the gating mechanism, but the carrier protein undergoes conformational change. Here you can see these gates these gates are open toward the extracellular space and closed toward the cytoplasm or intracellular space. Okay, in the next state, which is intermediate stage, after attaining the next stage, these molecules are closed toward the extracellular space and open toward the intracellular space, and will transfer or will transport the solute from the higher concentration toward the lower concentration until equilibrium is attained. So facilitated diffusion occurs through channel proteins or carrier proteins. Okay, channel proteins can contain ion channels or aquaporins. Okay, ion channels, basically these are highly selective. These are highly selective, like sodium, potassium, calcium channels. Okay, and these are most probably voltage gated. So voltage gated. Okay, also these are ligand gated, which means after the attachment of the ligand, this will uh, bring conformational change and will transport these whatever the molecule want to cross the membrane. It will bring the translocation. Then aquaporins are water specific channels. These are basically tetrameric proteins. These are basically tetrameric proteins which have narrow pores through which these water molecules are transported. Okay, aquaporins, for example, aquaporin example is aquaporin 1. Okay, aquaporin 1 is present in erythrocytes and proximal tubules of kidney. Okay, so let me write in erythrocytes or RBCs and proximal tubules of kidney. Then aquaporin 2, which is present in collecting duct. Okay, then we have carrier proteins. Carrier proteins will bind substrate on one side. This will bind to the substrate from one side. It can be the extracellular space or can be the intracellular space and bring conformational change. Okay, for example, the GLUT proteins means glucose transporter proteins. These are glucose transporter. These are substrate specific and bring conformational change and works on the conformational change mechanism. For example, GLUT1, glucose transporter 1, is present in erythrocytes. Okay, which will mediate the passive entry of glucose. In the same way, GLUT4 is present in the muscles and adipose tissues, which is recruited in response to insulin. So here you can see this is basically a channel protein through which these solute are get translocated toward the lower concentration. Okay, toward the lower concentration. This is higher concentration, and these substrate are most probably large polar molecules. Okay, then this is a channel protein. Sorry, this is a carrier protein. So these substrate molecules here you can see binds to the specific sites. Okay, then there is the intermediate stage, and then there is a conformational change in the carrier protein and it will translocate this molecule inside the cell. Then we have active transport, which will require ATP4 translocation, which will require ATP4 translocation. Let me tell you simple flow chart. Okay, let us consider there is a pump, or you can say a transporter. Okay, like this is a pump. Firstly, substrate will bind to this pump, then ATP hydrolysis take place, and this ATP hydrolysis will phosphorylate the pump. Okay, this phosphorylation will bring conformational change, and this conformational change will translocate the molecule. In this image you can also see, just wait, in this image you can see, firstly these molecules are binded, then ATP hydrolysis occur, that adds phosphate group, which will bring the conformational change and will translocate these molecules out, and the molecules which are present outside will translocated inside the membrane. So active transport is the movement of molecules across a cell membrane against their concentration gradient, okay, which will require energy input in the form of ATP. Okay, ATP hydrolysis will provide the fuel for active transport. The active transport employs specialized protein pumps and transporters embedded in the membrane that undergo conformational changes to translocate substrate from low to high concentration. Okay, active transport is essential for maintaining cellular ion gradient such as sodium potassium ion balance. We will discuss about the sodium potassium pump in the next slides. So active transport is basically of three types: primary active transport, secondary active transport, and vesicular transport. Primary active transport will use ATP hydrolysis directly, okay, and it will use the ATP indirectly. We will discuss this later. Then vesicular transport involves endocytosis and exocytosis. First, primary active transport. Primary active transport directly uses energy from ATP hydrolysis, okay, to move molecules against their concentration gradient across the cell membrane. It involves specific transmembrane protein pumps. We have already discussed that these pumps will undergo conformational change powered by ATP to transport ion or molecule from low concentration to higher concentration, and this type of transport is essential for cell volume regulation, nutrient uptake, electrical excitability in nerve cells or muscle. Okay, so here is the sodium potassium pump, which is a example of classic example of primary active transport. So let discuss it. Here you can see this is a pump. We can say it is in its even state, okay, in which it is opened toward the cytosolic side and closed toward the extracellular space. Firstly, the cytoplasmic sodium binds to the specific spots which are present here. Three sodium ions from the cytosol bind to the pump, then ATP will provide the fuel for this translocation, and this pump will become phosphorylated. Okay, we can say sodium potassium pump is phosphorylated by ATP. So after phosphorylation, what we have studied that phosphorylation brings, or we can say phosphorylation triggers the uh, conformational change. Phosphorylation bring conformational change. In this state, we can see this pump is in its E2 state, in which it is opened toward the extracellular side and closed toward the cytosolic side. It will release the 3 sodium ions in the extracellular space, and 2 potassium ion will bind to the specific site present over here, and after the attachment of potassium ion, phosphate group will leave this pump, and this pump will return to its original conformation, which is E1, and will release its potassium ions inside the cell. This is the sodium potassium pump. In short, we can say 3 sodium ions out and 2 potassium ions in. This is the sodium potassium pump, which maintain the equilibrium of ions outside the cell, and outside as well as inside the cell in the cytoplasm. In this animation, you can see 3 sodium bind to their specific site, then ATP will come, provide the phosphate group which will phosphorylate this pump,